Hi, good afternoon. Thank you very much for listening to the presentation. I will get right into it as soon as this works. There we go. As we are a publicly traded company, this is our safe harbor statement on forward-looking statements that we are required to make as we present our information. So this is a quick summary on Unique Eur, and I'll just say before walking through these slides, or rather these bullets, Unique Eur, as you may know, is a company that is Dutch originally, spun out of the University of Amsterdam, and is situated both in Amsterdam and in Lexington, Massachusetts. We have a large manufacturing facility in Lexington and our research capabilities in Amsterdam. This covers, it's easier to see it here, this actually covers a lot of the highlights of the company. First of all, we're developing our lead program, Hemophilia B, which is now in phase 3, and that is enrolling. It's finished its enrollment, and it's going into its, well, certain patients have been dosed, and the others are in their observation phase before being dosed. We are also have our Huntington's disease treatment, which is now IND approved and headed towards the treatment of patients. And we're working in several preclinical areas, some of which we've disclosed, and those are our SCA3 program, our Hemophilia A program, which we'll talk a little more about, and Fabry disease. In addition to these, we have, it's important to, I think, point out that we have global commercial rights to all of the programs that we are developing, with the exception of a couple of programs that we're working with Bristol Myers Squibb on in the heart failure space. And we have a leadership position in GMP large-scale manufacturing of AAV gene therapies, which will cover some details around that as well. And I'll talk a little bit about some of the technologies, including our gene silencing technology that we have called uniQure. So this is a glimpse of our pipeline. Again, the Hemophilia B program being in phase 3, and the various other programs a little further behind, depending on the program. We've organized it around liver targeting and CNS, and then our BMS collaboration. This summarizes our GMP Manufacturing Facility in Lexington, Mass. It is a large facility. We've expanded to now 80,000 square feet, and it is a third-generation insect cell baculoviral-based manufacturing platform where we have scaled up to 500-liter production, and we have the capability to scale up to 2 by 2,000 liter, a total of 4,000 liters, to upstream production lines at least at 2,000 liter, which gives us a very large capacity. And we have very strong intellectual property around our platform, which we'll cover a little bit later. And this facility gives us a lot of important capabilities that really have contributed heavily to our leadership position, one of which is the fact that it gives us, it's an end-to-end facility, so we can go from the earliest process and analytical development all the way through production and fill and finish and shipping of product directly to where it's going to be shipped. So there, we really use no third parties at all in any of the production. And this gives us enormous flexibility that's not easily found in the industry right now. In addition, the insect cell baculoviral platform is highly scalable, very cost-effective, and produces large volumes with a great deal of consistency. So this is a significant advantage of ours. We talk a little bit about our Av5 Vector, which is unique in several respects. These are listed out here. It has got a lot of clinical experience, and it is well known to have very low immunological implications, either in the form of pre-existing neutralizing antibodies or the creation of T-cell responses that can direct T-cells against the cells that have been transduced, and therefore the loss of effect of the gene therapy. This has not been observed in the long experience with Av5. So it's an excellent capsid for us to be using in our gene therapies, and we do use it in the majority of the programs that we have. I'll discuss a little bit the Hemophilia B program. I think very importantly is the phase 2B data in 3 patients that we've used and talked about the effect of our product. So what we're seeing is efficacy that is very impressive, an average of about 45% to factor IX activity level after however many months this is, about 30 months, 36 months, 36 weeks rather. And so the effect is very nice. It's industry leading. It is consistent and not highly variable. And we have data from our earlier programs that shows a long duration of effect. So this is an excellent program in hemophilia B. In addition to these qualities, the advantages of Av5 again also apply here. We have very low to no pre-existing antibody exclusions. In this study, we've seen no need for any immunosuppression whatsoever. The patients on this therapy have had no need to use replacement therapy at all. They've had no bleeds, and their factor level is very high and consistent. So it's an excellent program from that perspective. This next slide summarizes our phase 3 program. We have fully enrolled with 62 patients. And we are now, and some patients are being dosed, others are going through the 6-month lead-in process where we measure the bleeding rates and compare those to 6 months after they've been treated with the product to compare bleeding rates. And we'll be looking at factor IX expression, bleeding rates, the decrease in the need for factor IX replacement therapy, and of course, safety. So then I'll talk about our Huntington's disease program. So this has just entered the clinic, and it is a very exciting area, very high unmet need. And instead of the gene replacement approach that we're taking with hemophilia to give people a factor IX gene, we are actually transducing patients with the gene for a microRNA that then knocks down the expression of Huntington protein, which is the mutant form of that is deleterious and causes the Huntington disease. So this is a very interesting program in a very large market for an orphan disease with a very high unmet need. Part of what is important in the approach that we take is that we inject this directly into the striatum of the brain, which is number 1 on this picture, where we're able to fill up the putamen and the caudate. And then from there, because of the qualities of our Av5, it actually spreads through the caudate and the putamen and also up into the cortex, being in there, and we're able to see knockdown in both of these regions, these are the regions that are important to have knockdown in order to address the disease. And it doesn't spread to other regions. So this has very good coverage for the purposes of the disease. This slide provides some evidence from preclinical work to support the distribution into the putamen and caudate where the injection is actually delivered, but also in the thalamus and the cortex as well, where we do not inject. And then the following slide shows knockdown. So the lower the bars, that's a greater degree of knockdown. And you see significant knockdown in the striatum where we deliver it. You see also significant knockdown in the cortex and much less in other areas of the brain which don't need to be affected. So this has a very nice effect. We have additional preclinical data that has been presented in a lot of settings on efficacy in mice and other things that I think speak well to the potential for this program. The phase 1/2 trial that is currently initiated is, as you would imagine, it's clearly going to assess safety. And it's a multi-center trial where we will do a control group with imitation surgery and then a number of treatment patients as well in the United States. And we're targeting these early manifest patients with certain characteristics where we would anticipate being able to see some effect in a relatively short period of time for a disease like this. Next, I'll just cover some of the endpoints in the trial. there's both clinical endpoints as well as biomarker endpoints, imaging endpoints, and patient-reported outcomes. So we're going to look for motor symptoms, eventually cognitive symptoms as well. And then, as I mentioned, we're going to measure both brain volume and other aspects of that we can with MRI, and then biomarkers of NFL and mutant Huntington in the CSF to be able to determine and get some idea of what effect we're having in these patients. So next, I will cover our research pipeline. The first program in the pipeline to cover is our Hemophilia A program. This is different than other hemophilia gene therapy programs in that we are not replacing factor VIII, where the disease is a deficiency of factor VIII. What we are doing is we've developed, through a partnership, a form of factor IX that is factor VIII independent, although not activated, and becomes normally activated by normal mechanisms within the system. For those of you who are familiar or not familiar with what factor VIII does, factor VIII is a cofactor that brings activated factor IX into proximity with factor X and allows that activated factor IX to activate factor X. And what this does is allow that to occur without the need for factor VIII. So it's a very interesting molecule. The advantage here is several-fold, one of which factor VIII produced in hepatocytes, which is what the gene therapy programs typically do, has been shown to be problematic, and since hepatocytes don't normally produce factor VIII. So we are producing factor IX, or a variant of factor IX, in hepatocytes where factor IX is normally produced. So we expect that to be an improvement. And since this is not factor VIII, it is not subject at all to factor VIII inhibitors. So it can be used in inhibitor patients. It can also be used in non-inhibitor patients. And it doesn't interact with bypass agents. And in our early data, it shows very comparable efficacy to what emicizumab or Hemlibra shows as well. So in some ways, it can be thought of as a gene therapy version of that compound. In Fabry disease, we are also not just simply replacing alpha-gal, which is the missing enzyme in Fabry disease, but we've taken a modified NAGA, which is a different class of proteins, that has alpha-gal activity. And again, in this case, we see several advantages by taking this novel approach, one of which is that this modified NAGA has better uptake and bioavailability and other things than a typical alpha-gal. And remember, when you do enzyme replacement therapy, alpha-gal is normally produced intracellularly and works intracellularly, whereas when you administer something by either enzyme replacement therapy or gene therapy into the liver, it's got to get into the bloodstream and then into the target organs. And there is a significant degree to which that does not occur very efficiently. So this improves the efficiency and the efficacy. It will also work in patients who've developed inhibitors to enzyme replacement therapy alpha-gal. So it's, again, a very interesting program that occupies, we think it will be a very novel positioning within that market. The third program in our preclinical portfolio that we're describing is our SCA3. So SCA3 is in early development. SCA is a ataxia, and this is a genetic form very similar. It's also a CAG repeat disorder, just like Huntington's disease. So this really leverages a lot of our experience in Huntington. It's in a different part of the brain, and it's a CAG repeat in a different gene, the SCA3 gene. But it's very similar. We take our uniQure silencing platform, and we apply it against this target. And we've seen very nice effects for this in our preclinical models, and we'll be bringing this forward. It's got the potential to work in other CAG repeat diseases, including other SCAs. So that's a very interesting area. This simply summarizes our upcoming corporate goals, which I think was covered in the slides that I just went through. So this is what we expect to do in the near future. We will be generating top-line data in our Hem B program in 2020 with a BLA submission in 2021. And I think these others you can read through as well because I am now out of time. So thank you very much.